Hello friends, good evening. Welcome to Sam online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about various enzymes that are involved in the process of replication in prokaryotes. DNA replication is a biological process in which several enzymes work together to produce an identical copy of a chromosome. DNA replication occurs in all living organisms. In prokaryotes, several enzymes play different role in the process of DNA replication. So we are going to discuss about various enzymes that are important in the process of prokaryotic DNA replication. First of all, we shall discuss about the enzyme Dna Polymerase. In prokaryotes, there are 5 different types of polymerase. Dna Polymerase 1, 2, 3, 4 and 5. Out of these, Polymerase 1, Dna Polymerase 1 and Dna Polymerase 3 play a specific role in the process of DNA replication. So first of all, we shall discuss about Dna Polymerase 1. Dna Polymerase 1, it was discovered in 1956 by Arthur Kornberg. This enzyme is encoded by the gene known as Pol A. This enzyme consists of 1928 amino acids. It is a processive enzyme. That means DNA polymerase 1 catalyzes the addition of several nucleotides one after one at a time. That is known as processivity. Next is the enzyme Dna Polymerase 1 is having multiple domains. What are the multiple domains that are present? Now one is thumb domain, finger domain and palm domain. In addition to these 3 domains, there is a 4th domain and 5th domain. 4th domain possesses 3' to 5' exonuclease activity. 3' to 5' exonuclease activity is also known as proofreading. By its proofreading activity, Dna Polymerase 1 can remove the nucleotides that are incorrectly incorporated. In addition to this activity, the enzyme Dna Polymerase also possesses 5' to 3' exonuclease activity. 5' to 3' exonuclease activity is associated with 5th domain. So what are the functions of Dna Polymerase 1? The main function of Dna Polymerase 1 in prokaryotic DNA replication is the removal of RNA primer. So this is about Dna Polymerase 1. Second enzyme is Dna Polymerase 3. Dna Polymerase 3 is the primary enzyme that has high processivity, and this enzyme also play a very important role in the process of prokaryotic DNA replication. Dna Polymerase 3 was discovered by Thomas Kornberg, who was the son of Arthur Kornberg and Malcolm Zepter in 1970. This enzyme has high processivity. High processivity means the enzyme Dna Polymerase 3 continuously add a large number of nucleotides one after another before separating from the DNA template. You know that for the process of DNA replication, we need a DNA template. So before separating from the template, the enzyme Dna Polymerase 3 catalyzes the addition of several nucleotides one after one before separating from the template. So the Dna Polymerase 3 is having 3' to 5' exonuclease activity. 3' to 5' exonuclease activity is also known as proofreading activity. By this activity, Dna polymerase can rectify any of the error that has committed by this enzyme during the process of DNA replication. DNA polymerase 3 is a part of large complex known as replication. Replication is a complex that consists of different subunits which are present at the location of replication fork. You know what is replication fork? Replication fork is a Y-shaped structure that is present in the chromosomal DNA during the process of replication. So the replicome consists of two DNA polymerase 3 enzymes. Each DNA polymerase 3 enzyme contains one alpha subunit, epsilon subunit, theta subunit. In addition to these three subunits, other subunits are two beta subunits, two tau subunits, one gamma subunit, and psi subunit. These are the various subunits that are present in the replicome. First is alpha subunit. Alpha subunit is encoded by the gene DNAE. Epsilon subunit is encoded by the gene DNAQ. Theta subunit is encoded by the gene whole E. This theta subunit stimulate the 3' to 5' exonuclease activity of DNA polymerase 3. So DNA polymerase 3 is the primary enzyme in the process of DNA replication. The beta subunit, which is also present in the replicome, it acts as a sliding clamp. This subunit is encoded by a gene known as DNAN. The sliding clamp prevents the DNA polymerase to separate from the DNA template. Next is, it also contains two tau subunits. The replicome also contains two tau subunits. These two tau subunits play a role in dimerization. Tau subunit is encoded by the gene DNAX. The replicome also contains a single gamma subunit and other subunits that are present. One is chi subunit and the other is psi subunit. So these are the various components that are present in the replicome. All these components work together during the process of DNA replication in order to copy a DNA molecule into an identical DNA molecule. Another enzyme is helicase. Helicase is an enzyme whose main function is to unwind a double-stranded DNA. So the enzyme helicase open up the DNA double helix. This occurs due to the breaking down of the hydrogen bond. So the two strands of a double-stranded DNA molecule are separated. So various enzymes get access to the DNA strands in order to carry out the replication process. Helicase is a motor protein. Its function is to unpack the double-stranded DNA molecule. This helicase, it moves in a direction along the sugar phosphate backbone. So by moving along the sugar phosphate backbone, the enzyme helicase separate the two strands of a double-stranded DNA molecule. For unwinding the double-stranded DNA molecule, enzyme comes from ATP hydrolysis. In addition to separating two strands of a double-stranded DNA molecule, helicase also plays several roles. For example, homologous DNA recombination, in the process of transcription, in the process of translation, also in ribosome biogenesis and several other activities. So friends, this is about DNA polymerase 1, DNA polymerase 3 and helicase. These three enzymes are involved in replication in prokaryotes. There are some other enzymes also. In the next video, we will discuss about the other enzymes that play various roles in the process of DNA replication in addition to DNA polymerase 1, DNA polymerase 3 and helicase. So please watch my next video that will describe other enzymes that are involved in the process of replication in prokaryotes. Thank you friends. Please subscribe my YouTube channel Sam Online Class. Give your feedback. Thank you very much for reposing faith on me. Thank you. Bye bye.